This is episode 15 of the Immunology Podcast, Tuning the Immune Response with Dr. Anil Bhushan. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today we have Dr. Anil Bhushan from the University of California, San Francisco on the podcast to talk about understanding how the immune system senses and tunes its responses to senescent cells in disease and aging. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
looking to stay current with the latest research and news in the fields of immunology and cell biology, we'd like to remind our listeners to check out Stem Cell Science News, featuring the most recent top peer-reviewed research and review papers, as well as industry, policy, and science news. Stem Cell Science News provides a platform that allows researchers to stay up to date with their field while saving time. Subscribe for free at stemcellsciencenews.com.
All right, well, hello there.
Hi, Jason. Have you been following the latest Nobel Prize
announcements? Chiral chemistry using small organic molecules for organic synthesis.
Very interesting. I have to say, a little bit out of my scope of knowledge, but sounds fascinating. I cannot wait to read one of those nice overviews they give about the research they are awarding.
I may also be a chemistry major in a past life and have tutored and TA'd Orgo for many a year.
Why am I not surprised?
Yeah, but then also pain, so we talk organic chemistry. I know it causes a lot of people pain and sensation, and that was the Nobel Prize in medicine and physiology.
I will try to catch up to those research topics.
And I'll say bold choice, not going for COVID researchers, but I also think a wise one. So it's speaking to the larger world, and they can come back in a year or two once the pandemic is done, and then give the people we all know our due one.
Is it too early now? I think we're still in the middle of the thing.
That's the idea. I think it may be a little early. It's like a little too trendy. The Nobel Prize, they try to have the long view.
All right. Talking about long views and broad views of immunology, what do you have today?
Well, I don't know how long view this is given our current conversation, but we have, and it ties in organic chemistry as well. Here's a little segue, just because of the second word, the first word being A. So the title of the paper is A Prenylated Double-Stranded RNA Sensor Protects Against Severe COVID-19. In Science, first author is Arthur Wickigan. Last author is Sam Wilson. So this is an interesting paper, prenylation being, the post ribosomal modification to add a lipid group onto a protein. But they start getting into understanding the viral immune response in terms of like the other molecules we have. So not external factors, but factors inside a cell. And some of those are double-stranded RNA sensing proteins, which we have. One of those is called OAS1. And the way OSA1 works, not just for COVID, but for anything, is it will sense through binding, obviously, the presence of double-stranded RNA, and then induces RNase L, which then kills things. It kills the cell, that's infected. But what they found here that was very interesting, and so they did a whole bunch of work, and I can get into the mechanism in a second, but what they found is that OAS1 through RNase L inhibits SARS-CoV-2. Okay, so that's not that exciting because this is what that molecule would be presumed to do, or ones in its family. There's other OASs that they show don't do this. But what was really interesting was they found that there's actually two isoforms of this, which is known P46 and P42. P46, the longer isoform, has the marker on it to get prenylated. And it's only that one that works. And this has to do with that COVID is, or SARS-CoV-2, to be technical about it, is manufactured in perimembranous organelles. And so without the prenylation, this OAS is not targeted to perimembranous organelles to function. And so there's also SNPs in people, so single nucleotide polymorphisms, such that if you have the one that's one SNP, you make more P42 than P46, so you don't have prenylated. And that makes you more susceptible to COVID in patients. They find this. The susceptibility to COVID is really in relation to the amount of prenylated OAS1 you have. The more prenylated OAS1 have, the less severe disease you have. And that's just based on not just one SNP that makes you have more P42, but there's other SNPs that just make you not get prenylated, and they see that this correlates. And so it's really about the amount of prenylated OAS1 that can occur in your body limits your infection. And so this was a large amount of work, and it doesn't sound like it's going to be a lot of work to begin with, and then they just keep going deeper. They do a candidate screen with interferon inducible genes. They do this in a cell-based system, really map out the pathway, blockade various genes, so OAS1, but not 3, look at the polymorphisms, make mutations that can't be prenylated, map this all out in vitro, essentially, with culture systems showing that it works, and then they take it into people and show that it's really important there, too. And then they drop the gauntlet and get a science paper.
What do you mean they drop it into people?
So they show that severity of disease maps out this exactly. So that they can just take cases and sequence people, and the severity of disease correlates with that ability to have P46 prenylation or not. The more, and so that there's different variants in human, so that where P46 isn't prenylated, where it is, or if you're a P42 dominant SNP. And so they show that the less prenylated OAS1 you have, the more severe your COVID.
And does this prenylation also have some other effects on outside of the context of COVID infection?
It just targets something to the membrane. So it targets this OAS1 to the membrane. So other diseases where OAS1 detects viruses that are not replicated in a perimembranous replicative organelle, this doesn't matter.
So it's very specific for coronavirus.
Or for other viruses that happen to have the same replicative structure. And they go into animals and show that there is an ancient retrotransposon that ablated OAS1 prenylation in the horseshoe bats, which is where coronavirus, SARS-CoV-2 comes from.
Cool.
So they lost the ability to resist this virus.
I wonder if this, now, thanks to modern science, we have the chance of saving the lives of a lot of people infected. But I wonder if this had happened in some kind of ancient situation with no sufficient medicine, then would that generate some kind of gene drive towards one of these?
Oh yeah, it would select out the SNP that makes more of the non-prenylated version. Absolutely.
Very interesting.
Oh yeah. So they link this down to, as I said, they really go deep, and they show that this applies to MERS and other, similar viruses with similar replicative features, but not viruses that don't replicate similarly.
Very nice. Well, now it's my turn, and I have a paper to share with you that I think you'll pretty much appreciate. It has a lot of things you like, so let me just get on with it. Title: Low Neoantigen Expression and Poor T cell Priming Underlie Early Immune Escape in Colorectal Cancer. First author, Peter Westcott from the lab of Tyler Jacks at the David Koch Institute at the MIT. And very interesting work. I personally enjoyed it very much. They are looking into, as I mentioned, colorectal cancer. So we know that colorectal cancers can be roughly divided into two groups that are very kind of important to their prognosis and their treatment, which is microsatellite stable and microsatellite stable colorectal cancers. And microsatellite unstable cancers are usually characterized by a higher tumor mutational burden because they have a more, a less stable DNA, so to say. And the enhanced mutational burdens in the unstable colorectal cancer usually relates to a higher expression of neoantigens, and often patients respond better to immunotherapies and checkpoint inhibitors than colorectal cancers that are microsatellite stable. And so the authors were looking into what other factors are also affecting the capacity of the immune system to detect and to fight these tumors, these colorectal tumors, because what they see is that although they express less of them, microsatellite stable MSS tumors are expressing some level of neoantigens, and they do have some amount of mutations, which would potentially be able to be picked up by the immune system and by T cells. So they look a little bit closer into the expression of neoantigens in human samples of colorectal cancer from the Cancer Genome Atlas, and they start doing some kind of fishing and looking into previously generated data, and they do see that in fact, as expected, the MSS tumors have a lower mutational burden and have a lower amount of neoantigens. But what's also interesting is that not only they have less different neoantigens, but they express the ones they have, they usually expressed at a lower level. So there's these two things that characterize the neoantigens in MSS tumors. And they found that for all of the samples that were analyzed in this data set, all tumors are expressing at least a couple of clonal neoantigens, so neoantigens that are expressed in the whole tumor. So their question is, what is going on, and why are these neoantigens not being picked up by the immune system? So they try to make a mouse model in which they have an antigen, a model antigen of albumin, and they are trying to induce colorectal cancer in mice. So they inject in the colon of mice lentivirus that are expressing CRE, and they're using mice that are flocks in APC, which is the knockout of APC is fairly common amongst colorectal cancers. And so they see that if they induce this and they have overexpression on top of this knockout, they see that indeed the few tumors that are developed, tumors are not developed as much. So there's an immune response against this over antigen, but those tumors that develop lose the antigen, and therefore the authors find themselves thinking, this is not going to work for us, because we want to see what happens, how the different antigens are affecting the immune response. And they decide to move out of injecting this CRE lentivirus with CRE, and they actually move into injecting organoids. And they have an organoid which is quadruple mutant. They have an APC knockout or knockdown, SMAD4 knockout, KRAS mutations, and TRP53 knockout as well. And they include the expression of albumin, but they have different variations of this vector in which they end up expressing OVA at different levels. And this is the key. They end up having different cell lines that express OVA at different levels. And they have a high expressing cell line and a low expressing cell line, and cell lines kind of in between. And when they use this, and when they inject this organoids into the colon to start a tumor in the mouse, they see that the high expression of OVA, the high expressing cell lines, tumor organoids, really end up being rejected by the mice. But those who are expressing OVA but at low levels, the tumors grow as in the no antigen, the no OVA controls. And they see that this happens. This happens really is very important. The early time, it is very important how the expression of antigen is in the early time after the injection. And they see that already early in the early time points, the antigens that have a low expression of the tumors have cells that already have a diminished effector function, and they start very quickly to show signs of dysfunction much faster and much more clearly that those T cells are infiltrating tumors that have a high expression of antigen. And so what they see, and kind of a long story short, they find that for them, what is very important is the expression of the neoantigen really has a impact on the priming and on the start of the T cell response. And they pinpoint the importance of these tumors that are expressing a low or a high amount of antigen being picked up by dendritic cells, and these antigens being presented to T cells, and the intensity of the presentation depends on the level of expression of the neoantigen on those tumors. So under this logic, they see that if you have tumors that have a high expression of antigen, but are actually knockout for MHC1, they can still prime the immune response and and generate T cells that can pick up and kill tumors expressing a low amount of antigen. So it's not about the antigen expressed by the tumor, but about the antigen expressed by the cells that are picked up for DC presentation and priming of T cells. And so they think about this in a more in vivo. So there are other in vivo experiments in which they find that therapeutic priming with neoantigens and also anti-CD40, which really activates antigen presenting cells, can be very beneficial and can mediate the priming of the immune response, even in the situation of low antigen presentation. And it really makes a difference and synergizes when it's combined. Anti-CD40 treatment, for example, is combined with immune checkpoints such as anti-PD-1. So they also looked they also have some so the basically what they do is they start with looking at the human situation, in which they find that the level of expression of these neoantigens kind of also predicts the outcome of the outcome of the response. And they end up making this really interesting mouse model in which they can have different levels of expression and evaluate specifically how that affects the immune response.
Whew. All right. Well, so does it sound like then that it's about the dose, right? So high levels early on induce a stronger primed response that in the immune cells that then induces a kill effect where low levels generates tolerance. And so then hence checkpoint inhibitors and everything else will shift even lower levels to a kill response versus a tolerance response.
Exactly. That is, and I think that's a very now widely uh, spread idea that this initial priming, if the initial priming is very poor because there's not enough inflammation, because usually, tumors start small, and that can really lead to T cell dysfunction for these T cell clones that get activated under these conditions.
Excellent. All right. Well, I'm gonna flip here and also talk about the colon. Shock of shocks.
Shocking.
Yeah, Well, when I saw this had popped up, it was a good one. It's an immunity. It is cognate recognition of microbial antigens defines constricted CD4 positive T cell receptor repertoires in the inflamed colon. So its first author is Moritz Muschewek, and last author is Oliver Patst. So this is one of those things where we already probably thought we knew this, but no one had actually shown that we know this. And that's actually why it's important, is because really trying to understand some in-depth biology can get hard. So this entire paper is underpinned on using a T cell model of colitis, a T cell transfer model. So you take a rag knockout mouse and you give it T cells, and then it develops colitis because all these T cells are not used to being, even though they should be, genetically very similar. The T cells freak out to the novel microbiome and then give you colitis. So what they see is that they basically did this a whole bunch of ways and sequenced the TCR alpha repertoires to understand what was happening in different scenarios of this. So they actually map out the T cell receptor repertoires, and then they show that they can, by various levels of sequencing as compared to more in-depth techniques, they can find that, yes, a quick sequencing can give you an understanding of these different repertoires and map out how clonal they are. And so to kind of dive in, what they did is they found that when you just do this generally, you get different repertoires. So if you just take a whole bunch of mice and transfer the same pool of cells to different mice, you get different clonal expansions in the different recipient mice of their T cell receptors that are and they look specifically interferon gamma and IL-17 producing cells. But you get these dominant clonotypes were shared between these two different types of cells. They weren't able to do double interferon gamma IL-17 producing because of limitations in flow, and then double mapping them and everything, and then sequencing. They just couldn't get that resolution. But this doesn't happen with FOXP3 regulatory cells. identical clonotypes accumulated in the colons of different people, of different individuals. In this case, it was mice, right? But so if you see clonal expansion and you see the same types of clones pop up across individuals, but if you give antibiotics to ablate different subspecies, that effect goes away. Basically, if you window down either by having a definite colonization, so you put in a specific bacteria that's not normally there into all the mice, or you ablate a whole bunch of bugs with antibiotics. If the antibiotics have a strong effect on the microbiome, so they sculpt in a consistent way, or again, you put a bug in, you get the same clones out. In that case, it becomes more distinct patterns appear. And so then you get the same thing. And then they did this really neat thing where they clonally expanded the cells before putting it in. And they saw that this, if you just take different cultures, again, if you take different naive T cells and you expand them out and you put them in to different mice, or if you take different T cells out and just pop them in, you get different clones that appear and everything. But if you clonally expand first, if you take cells, clonally expand them, and then pop them all into different mice, so right, the same clones all go in, a pattern emerges. And so the same clones then pop up. So basically, clones going in equals clones going out on the T cells. And that's what they really found, is that you basically have this pool of, unselected for T cells with different TCRs, which we know, right? They're hanging out. And then which one wins at baseline is kind of a crapshoot. So you're going to have different clones that pop up in the system of induced colitis. But if you take a pool of cells and massively expand it, and then introduce that clonally expanded pool, you're already biasing that cell through the clonal process to select for specific clones more than others. And those dominant clones then reemerge, some subset of that, when you then put it into a microbiome background. So when you put it back into a mouse, it develops inflammation. You see these TCR clones populations repopulate. So you see the same clone appear in all the different mice as being a dominant clone. And so they're really showing that in the end, you kind of, the way it's working in the gut is that you have these two things. Different microbial species have dominant effects on the T cell repertoire. So specific bugs do it, other ones don't. So they show this by, if you give vancomycin or a specific bacteria that's not normally there, you see it really hone in on that, the T cell repertoire. But if you give something like an aminoglycoside, which doesn't do this as much, it doesn't sculpt it in the microbiome the same way, you don't get the same flag raising, these same clones keep popping up. But it's the donor T cells. It's the T cells that you start with that defines the repertoires that pop up. So the same identical donor, the recipients transfer the same identical cells, have similar TCR alpha repertoires. And that's what was very interesting. And then this also then goes along with the level of colitogenicity that goes along with it. So certain T cells are going to be more cholitogenic. And so if you home in on that, you can then drive the disease by having that clone that's more cholitogenic past all the people. You're going to see similar colitis. If you have a more heterogeneous set of clones coming in, you have more heterogeneous levels of disease.
So how far away are we from the magic pill with the right bacteria to get the right T cells?
Oh, yeah. A while.
Looking at you.
I know. So I think the thing is, how would you switch the bacteria over? Everyone's coming into the game with different bacteria and different T cells.
Yeah.
So they do talk about how, this could, help understand diseases and stuff. But I think the real point is, if you targeted dominant T cells, that's their point. If you target dominant T cells that are causing disease, if you deplete those, that's how you get rid of disease. Mm. Versus resculpting the microbiome. You'd have to transplant them away from bacteria that are objectionable. But what bacteria are objectionable to a T cell depend on the T cell clones in that host.
Do they say why they are sequencing TCR alpha chains? Because it's usually the beta that gets
So they show that alpha and beta, they did both at the beginning. And they found that they had better data with alpha, and that either doing alpha or beta predicted it, but that was less noisy with alpha, so they ran with alpha. They actually validated that first off.
Okay. Interesting.
So they wouldn't have to double sequence. They basically did both in a subset, showed they had better reads with alpha, and then ran with it.
Okay. Fair enough. Okay. Next, moving on to my last publication of the day. So this publication comes from the labs of Miles Brown and Shirley Liu at the Dana-Farber Cancer Institute in Boston. It's a bunch of first authors, and it was published in Cell. And it's titled In Vivo CRISPR Screens Identify the E3 ligase COP1 as a modulator of macrophage infiltration and cancer immunotherapy target. And that basically gives away most of the work. So in this case, we're moving from colorectal cancer to triple negative breast cancer, which is also another cancer that is famously refractory to immunotherapy. It's a very complicated cancer to treat. And in this case, the authors are paying kind of a more close attention at the microenvironment and the tumor microenvironment, because they also see that that is probably the reason why this immunotherapy is not working, is because there is something in the tumor microenvironment that is really getting in the way of the immune response. So what they do, as many people have been doing lately, they start with a very nice CRISPR knockout library that is targeting many genes that are related to cell proliferation, immune function, and signaling. They have a very comprehensive kind of coverage of all these 4,500 genes, 5 single guide RNAs per gene. And they use this to modify 4T1 cells, which is a mouse triple negative breast cancer cell line, which is also expressed in ova as a model antigen. And they transfer this, so they modify, they transduce this 4T1 cells expressed in ova with this lentiviral library, and they inject these cells into the different mice. They have nude mice, which, as they lack thymus, they don't have T cells. Those are, they lack T cell responses. They have wild type bulb C, and they also have bulb C that had previously vaccinated with ova, so they're kind of primed mice. And they show that so they follow the growth of these tumors, and of course, what they see is that nude mice are the ones who perform the worst. So they have the highest tumor volume, and pre-treatment with ovalbumin in wild-type mice also reduces a little bit the tumor volume. So they show that there is a level of immune response against these tumors, but it's not enough because the mice end up dying anyway. So what they do is they look into, so they have these mice that have a knockout library, and they look after 16 days after the tumor challenge, what are the differentially expressed genes in this tumors that are left after, that have grown after this amount of time. And they see they identify a series of genes that are differentially expressed. And just kind of long story short, they identify one particular one, which is the COP1, which is a ubiquitinase. Ubiquitinase, exactly, that's the one. And that is really one of the most one of the genes that are most pop up in this library. And they do a second round with, they chose the highest, the best hits from this initial library with 4,500 genes, and they do a more focused library of those most promising genes. And they show that by knocking out these genes, again, they end up having lower tumor volume, so that the tumors grow less, and they see more T cell infiltration. And again, COP1 really pops up as one of the most interesting targets, and really is the most significantly depleted gene in these tumors. in the immunocompetent mice. And because then they really compare the immune competent mice versus the nude mice, in which the absence of COP1 does not really affect the tumor viability in vitro or in nude mice that do not have a functional immune T cell response. So they follow up on COP1 knockout, and they're trying to understand what is COP1 doing that is mediating this effect, this immune in the immune response. And what they see is that those knockout COP1 knockout tumor cells are expressing significantly less key macrophage chemoattractants and cytokines that are involved in macrophage activation and recruitment into the tumor. And they also, when they look at the tumors, they see that there is a decreased macrophage infiltration in this COP1 knockout tumors, and without significant changes in other immune cells. And they really see that this is most likely the mechanism by which, in the end, COP1 knockout is mediating a reduction. So the loss of COP1 results in less macrophage recruitment, and therefore less tumor escape, because usually these macrophages are this kind of known as M2, or this tumor-associated macrophages that really get in the way of the immune response more than anything else. So they look a little bit into the mechanism, and they see that the most important target of COP1 that pops up from their analysis is CEP delta, which belongs to the CEPB family of transcription factors that regulate many biological processes. But in this case, the absence or the overexpression of these genes ends up encoding these macrophage attracting chemokines and results in a higher tumor growth. And I think what is very interesting is that when they look into human samples, they go to the Cancer Genome Atlas. These guys do it as well. And they look into breast and colon cancer tumors, and they can see that in this data, they pick up that the COP1 expression is correlated with M2 macrophage signatures in this tumors. They see that the expression of the target of COP1 is negatively correlated with this M2 signature, and they see a positive association between COP1 protein and macrophage-associated cytokines in the samples, including, for example, CCL2, CCL7, and other chemokines in this category. So they see also a correlation between COP1 expression and the survival of affected individuals in these cohorts. So it kind of they really make this very compelling case towards COP1 activity being related to macrophage infiltration also in humans, and thus affecting survival and progression of disease also in humans.
So my question is, how druggable is this? Because the instant you start doing an E3 ligase, you're messing up everything and its uncle.
Yeah, that's also, authors also mentioned that it's a hard target to drug, so they don't have a suggestion, and they also don't know what drugging, like in this case, they have only the tumor being affected by the knockout, so it's not clear what would happen if you put some systemic drug. So I think they are a little bit, at the moment, they don't have a drug they can use.
Yeah, that's always the hard part. Find a cool thing, but if you get rid of it, it doesn't work for you, the rest of you.
Yeah. I think it was an interesting model. It was always is nice to see when the hit comes from one of these big screens. It's nice to see how they work on it quite well.
Yeah, and then that does it work. I mean, people forget there's all types of things that are completely lethal to cancer, but that are also lethal to the rest of you.
Well, but I think what is important is that this, as you said, this COP1 would have not been picked up in an in vitro, for example, screen. This only works in the in vivo situation. I think it's also important to keep in mind that often a lot of drugs are tested with kind of in vitro analysis, and then if you don't have, in this case, you're not evaluating the effect of the immune response, which is so critical for this particular target.
Yeah, so in this case, it's probably going to work, but it may not be druggable.
Yes.
All right. Well, we're going to be speaking to Dr. Anil Bhushan at the University of California, San Francisco in just a moment. But before we get to that, explore scientific resources for your immunology research at the STEMCELL Technologies Immunology Learning Center. Choose from different research areas and find expert interviews, technical tips, educational webinars, instructional videos, and much more. Visit stemcell.com/immunology-research.
Before we get into the episode, we'd like to give our listeners a heads up that Jason is not in the studio today, but he's cutting into the interview while traveling. So you may notice a difference in the quality of his audio, and we're sorry about that. Now back to the show. Hi, everyone. Today we're joined by Dr. Anil Bhushan. He is the professor at the Diabetes Center of the University of California in San Francisco and also co founder of Decidius Therapeutics, a company based on his research. He's joining us today from San Francisco. We're really happy to have you today talking to us. Welcome.
Thank you.
So, a lot of your research is around the impact of senescence and senescent cells on the organism and the role that the immune system has in clearing the cells from a healthy organism. And I think many immunologists might not think a lot about senescent cells and senescence in general. So maybe we could start this conversation with you telling us a little bit more about what is senescence and what is the role of the immune system in managing these cells.
Yeah, so that's an important question. I mean, we are unfortunately limited by the word senescence associated with aging, but really, senescent cells are damaged cells or cells that are somehow aberrant that need to be recognized by the immune system and removed. I mean, there's four major triggers of senescence that we know of today. One is a DNA damage induced senescence because of injury or because of other metabolites or something that damaged the DNA in the cells that then release the DNA exogenously in the cell, which then is recognized by the immune system and removed. And this happens in many chronic diseases, which we'll get into. The other major way the cells become senescent are what's called the oncogene-induced senescence. These are senescent cells that are turned on because of tumor cells. And the third major category is the viral induced senescent cells, and this is more recently shown in a couple papers. Is it's been shown in the last 5 years, but more recently, just last week, in a couple papers on SARS-CoV-2 and leading to senescence in the epithelial cells that it infects. And again, it's the same way the immune system recognizes these infected cells and removes them. And the fourth way is radiation. Radiation also leads to senescent cells, and again, the immune system recognizes them and removes them. So these are the four triggers of senescence. And senescent cells are basically a response to these triggers, and they exit the cell cycle, upregulate some classically cell cycle genes that are exit cell cycle inhibitors that are upregulated, and then there is a DNA repair going on if it's a DNA damage, or various repairs for the cells to happen. And when there's a decision that the cells cannot fix the problem, they turn on what's called a secretory profile, or otherwise known as SASP, senescence associated secretory profile. And this is what then communicates with the immune system and leads to its own removal.
So I like naps. What's wrong with a senescent cell? So we talked about how we get there, but I think it's important for the audience to understand what's wrong with them. Why shouldn't we have senescent cells? Why does the immune system need to clear them versus just having some cells at rest? That sounds good to me on a Sunday afternoon.
Yeah, especially if you're watching television. Uh, no, so the reason these cells are are essentially they create havoc in their microenvironment. So they have a secretory profile that includes growth factors, extracellular remodeling, extracellular matrix remodeling enzymes, uh, cytokines, chemokines. I mean, it's a whole host of over 60 factors that a secreted senescent cell releases. And that really it has a lot of non-cell autonomous effects, which meaning that they affect the local cells, not themselves necessarily, but other cells around them. And they generally create havoc where they are. And part of the reason evolutionary this whole thing is designed is so that it then communicates to the immune cells around it to recruit them, make a way for them, almost essentially like the construction workers. They build a highway for the immune cells to arrive to them, and then they also have flags to say, "This is me, get rid of me." And so all of that process is evolutionarily conserved in some sense in wound healing, for instance, or other, during development. And so their general way is that they're creating havoc to get rid of themselves. And once they're removed, then there's renewal in the tissue. And if they hang around, then they just create a lot of havoc, and that's how a lot of diseases come about. That's our that's our not hypothesis, but that's our essentially the basis of a lot of our work.
You just mentioned senescent cells in the case of COVID and COVID-2 infection. Where else do we often or we have senescent cells that are causing maybe disease or particular places where these cells are found?
Yeah, so the most often, senescent cell. So these cells form. I should make that very clear. The senescent cells form probably all the time. You never see them because when you have an efficient immune recognition and removal, then they are removed and it's never an issue. The only reason you see senescent cells accumulate in a tissue or in a body is when your immune system fails to remove them. So that was the key conceptual recognition that came in our lab about 3, 4 years ago. And so the idea is that this immune surveillance system that is built in the body naturally is failing to recognize these senescent cells, removing them, and that's when you see accumulation. The most often tissue in humans that you see accumulation is in the adipose. So in the fat tissue, in the white adipose tissue, there is a lot of it's heavily immune infiltrated as well, but there are a lot of progenitor cells. These are mesenchymal origin progenitor cells that give rise to a lot of um, cell types within the adipose, as well as they have the ability to give rise to many other cell types like bone and others. These are these stem progenitor cell-like population, and they tend to get uh, they become senescent, and we can go into reasons why. And once they become senescent, there is the general progenitor population for the adipocytes is reduced, and so that's typically one common area where you see accumulation of senescent cells.
So, on this topic, so you just recently published a paper in this journal from Cell called Med. I think it's a recently launched journal, and in which you are looking particular in one subset of immune cells, which are invariant NKT cells, and that can find there's one particular target that they recognize on senescent cells. Would you like to tell us more about this, the work that your lab did on this regard, and maybe whether there are other immune cells and other targets that are also involved in immune clearance of senescent cells?
Yeah, so the way we landed on this is what we did is we mined our single cell RNA-seq analysis of senescent cells. Um, and when we look, this allowed us to one, categorize senescent cells separately because there's no real actual method to really look at senescent cells by flow cytometry. You can create some enrichment methods, but there's no real way to there's no marker for senescent cells that you can use to and to isolate senescent cells. So what we did is we enriched for senescent cells, and then we based on transcriptional profile, we could look at senescent cells in a deep fashion and compare it to other cells. And what was striking and really obvious was that one of the big things that was changing in senescent cells was the antigen processing machinery itself was altered. And when we looked at the antigen processing machinery, which consists of a lot of MHC class one molecules, but also the top consistently the top uh, upregulated gene was what's called beta 2 microglobulin, B2M, which is an architectural protein that supports all the MHC class 1 molecules. So we said, oh, well, there's something going on there. So let's identify which MHC class 1 molecules are upregulated in senescent cells. We couldn't see that in the RNA-seq, so we designed a simple screen where we took these progenitor adipocytes from mice, and then we could culture them in vitro and make them senescent by treating them with chemotherapy agents to make them senescent. So it's a very classical way to do in vitro senescence. And with that, we could then screen for all the MHC class 1 molecules we could think of. And one of them stood out right away, which was a MHC1 class 1 like molecule called CD1D. And that was the key essentially to the lock that we had, because CD1D it only communicates with the iNKT cells. And the way it does is it presents a lipid antigen. Unlike most other MHC class 1 molecules, which present peptide antigens, CD1D presents a lipid antigen. And CD1D lipid antigen bound, a presentation recruits and activates the iNKT cells. So that's how we got into the identification of the iNKT cells. I should mention again, the iNKT cells are a subset of T cells that have some characteristics of the innate immunity like NK cells, but they have, like all T cells, they have a TCR, T cell receptor, which is, specifically is in the VDJ recombination is mostly invariant, so the alpha chain is always the same. And it recognizes the lipid antigen bound to CD1D, and that's actually how we isolate NKT cells or identify them in flow cytometry is using what's called a tetramer, which is a CD1D bound to a lipid antigen, and that's how we identify them.
So, to take this a little higher level, there's too many senescent cells in adipose tissue. I assume America, that may have something be related in some way to America's growing BMI, for example. What happens if you can pull the trigger, like you show in some of your research, and you get rid of them? What does that mean downstream for patient health?
Yeah, so I mean, this is where it becomes interesting. So here we have this mechanism that we elucidated. The question is, how could we therapeutically harvest this? Um, and um, so the way it works is that if we designed a simple mechanism where what if, first of all, we look to see, the senescent cells are accumulating in the adipose tissue in the mice as well as in humans. We figured we just characterize that, which is just a descriptive issue. But more importantly is when we look at the iNKT cells surrounding. So the iNKT cells, unlike most, these are resident iNKT cells. So the adipose iNKT cells live there. They take on their own identity, living in the adipose tissue. They're like immigrants in a country. They then come. They have their own Thanksgiving and everything. So they live within the adipose tissue, and um, they when we isolated these iNKT cells from compared them to adipose tissue with senescent cells or without senescent cells. What turned out to be one is that they are reduced in numbers. So first of all, you could tell right away that the numbers are like almost half. And this was not really, coming necessarily from our lab, and it was gratifying. I mean, we could confirm that, but it was initially described by a lab in Boston, Lydia Lynch's lab. And so these were, highly reduced in numbers, but also they were less active. They had less activation markers, like what's classically called CD69 or others. They were essentially what we would call anergic, sitting and unable to function. And so we said, okay, can we then reactivate these cells, and then with that remove the senescent cells? That was the crux. Can we, if we can do that, then we can certainly make this more therapeutic. And so we delivered a lipid, classical lipid antigen called alpha gal which is a microbial derived lipid antigen that is specific for activation of iNKT cells. And upon delivery of these lipid antigens, within hours, you could see the activation of iNKT cells, and that's classically, the cytokine release profile that's typical of iNKT cells, and you could measure that in the blood, so it's a good biomarker for activation. And also 4 days later in a mice, we could then look at the resident iNKT cells in the adipose tissue and see, one, that we have a great expansion of the numbers of these cells. So they went up to 10 to 20 fold and expanded, as well as they clearly have activation markers now active. So we could activate the iNKT cells. Then we looked at the senescent cells in the adipose tissue, and we could see that they are now depleted. So that was how we connected and shown that activation of iNKT cells in a, let's say, in a model, in a high-fat diet model in a mice where they accumulate, could be removed by activation of iNKT cells.
And then I guess my question therapeutically is, let's say we can do that. How do you only activate those and not turn on your body's entire immune system and then give, cytokine storm or systemic immune activation syndromes or other
Right,
other problems that we see with, say, immunotherapy is a
right.
common example?
So the fortunate part of this is there's two things fortunate. Well, I should also say there's two parts to this. Removing senescent cells was not just academic, but we can go into this later. But it was had a physiological effect on the disease that they cause. But we'll get into that later. But the question that you asked, which is a very valid question, is, are we activating a lot of general activation of the immune system leading to a cytokine storm? I mean, so there's two things that are worked in our favor to some extent. One is that the percentage of iNKT cells within the body is so small; they make up an amazingly small fraction, between zero point 0.1 to 0.5% of the cells. So if these cells start to secrete cytokines, it's still a very minuscule amount compared to activating, cytotoxic T cells or others, which make up a large fraction of the, lymphocyte population. So cytokine storm wasn't such a big issue because of its small numbers. Two, because of a surveilling system, there is a natural turn off of this activation. So it's a very transient activation. So we activate with a lipid antigen. These cells respond and release their cytokines very early on and then proliferate. And then there's a natural shut-off mechanism. The TCR of these cells is endocytosed, and they undergo apoptosis, and the numbers are back down. So in a mice, within 10 days, the numbers are back down to the low levels that we have seen before. So clearly, this is a transient activation, and we limit the amount of secondary activations. But it is true that the iNKT cells can activate other cell types as well. But we haven't seen any sustained activation of other cell types around them, or systemic changes that allow So this seems to be a very transient activation.
Talking again about what are there any diseases that are characterized by an accumulation of senescent cells, or is maybe that it's not only aging, is aging a consequence of accumulation of senescent cells, or what kind of diseases could we treat, for example, by being able to selectively deplete these cells?
Yeah, so every month we seem to find a new disease that has accumulation of senescent cells. I mean, this is a very active field, and a lot of labs have been showing a lot of diseases that are due to the accumulation of senescent cells. So there's a couple labs at the Mayo Clinic that have generated genetic mice models where they keep showing, in many different disease models where they can deplete the senescent cells and show efficacy, primarily from James Kirkland's lab and from Darren Baker's lab. And these are people that have been showing accumulation of senescent cells in many different diseases, and to just, to name a few, what I hinted on was, type 2 diabetes, but also, that arteriosclerosis is another example, the chronic kidney disease, and the list goes on and on. We've shown for type 1 diabetes, which is even in pediatric patients, in many neurodegenerative diseases, and in some cases and of course in a lot of cancers that senescent cells play a role in its progression. So the problem in this field is not that there's not enough diseases to go after with senescent accumulation. The problem is it's a plethora of diseases, and the question, especially when you're starting, a company that needs to focus, a small company, a big part of the time is spent on indication selection.
Before we move to the work you have been doing with your company, bringing this uh, a therapy to the patients, I just wanted to ask you a little bit more about so what do we know about invariant NKT cells? Do they only so far are they only have been found targets that are this kind of lipid targets, or are there other antigens that are these cells are also recognizing?
So there's different classes of iNKT cells as well. So there's, the class 1, class 2, and class 2 is not really what we're targeting. They're not truly invariant, they're semi-invariant. And they are involved in some autoimmune diseases. And they actually, I mean, their activity is needed and important. So we need to make a distinction between class 1, class 2 iNKT cells, or NKT cells, I should say. The iNKT cells are typically class 1 NKT cells. And they play multiple roles. They're generally roles in recognizing foreign cells, whether they're microbial antigens, which is typically of bacterial origins, which happens to be a lot of lipids that expressed in these microbial. Um, so they really are a surveilling system to protect us from invading pathogens, as well as essentially recognizing foreign cells that shouldn't belong to us, which is the flip side of autoimmunity as well. So that's always there.
So I wanted to flip into your biotech deciduous your startup that you've been working on, which presumably is to help get some of this to that next level, which means eventually patients. Without stepping on anything you can't talk about, when you think about talking to people and explaining what disease targets you're going after with like presumably these lipid antigens, what comes to mind first? What is the thing that you're going after to investors or just on the road into biotech going, hey, this is this is what this can be for right now based on what we know today. As you said, lots of diseases keep showing its importance, but there has to be
Right.
something at the top of the list that drives
Yeah.
that
So
now.
so first thing to recognize, and part of it is to educate the investors, is that this is a new way of treating disease. The classical way of treating, let's say, I give you an example of type 2 diabetes. Type 2 diabetes is treated by saying, oh, you have high blood glucose. We need to control the glucose. And so the treatment is typically focused on either the non-functioning of the pancreatic beta cells or the ability to metabolize glucose and get rid of it. So that's where all the treatments, that's the traditional pharma approach, the Novo Nordisk, Merck, Eli Lilly, and that's what they have focused on. What we are trying to do, for instance, and I'm just using type 2 diabetes as an example, is that this disease is not focused on one cell type or one organ, but it is actually in multiple organs, and what they call complications really come from the fact that accumulation of senescent cells occurs in multiple tissues in this disease state. So in the liver, you have accumulation, and there's a lot of correlation between, non-alcoholic liver disease, fibrosis disease with type 2 diabetes. There's also the chronic kidney disease that comes associated with type 2 diabetes, as well as I mentioned, arteriosclerosis and cardiac fibrosis. So, and all of these tissues show accumulation of senescent cells. So when you are systemically removing senescent cells, you're actually treating a disease holistically rather than looking at its symptomatic outcomes and treating it that way. So this is almost a paradigm shift in how you treat a disease, and this is what we need to educate investors on how different this is from previous efforts, and how in a holistic way that we're trying to treat a disease. So, but given the question that you asked, is given the fact that there's so many diseases that have this issue, we as a company, as a small company, you need to focus on some initial targets that are doable. A type 2 diabetes trial is outrageously expensive, and you can't really do it without major pharma buy-in. And so the idea and to prove the efficacy of these diseases, the plan for deciduous is to do a clinical trial in a phase 1B, sort of a basket trial where you have multiple indications, patients that you have, this will not be in any way exhaustive, but at least it gives you indications of multiple indications that you can choose to enroll patients in, and then decide on which phase 2 trial you can go into that's feasible given the money that the company will raise to do it. One example, for instance, is idiopathic lung fibrosis, which is a small population, but it's more manageable to do. And then, there's chronic kidney disease that is also manageable in some ways. So those are the kind of things that you look for.
How has been your experience so far moving into the biotech industry, trying to transform your kind of academic research into something that can be presented to investors and that can actually be developed into a treatment option?
Yeah, I mean, it's part of my evolution as a scientist, as a person as well. I mean, I started my career in science studying absolutely very basic issues of cell differentiation in early germline formation. And, at some point, I would have liked to see, translation into disease areas. And when this opportunity arrived where I could see a simple, not a simple, but at least a therapeutic pathway, I jumped at it because I wanted to, translate our ivory tower thinking into actual benefits to the human population.
That is quite fascinating, and I look forward to see how you can if you can reach clinical trials and whether your therapy shows some promising results. It's been really interesting talking to you about your research, and I think that before our conversation, I was really interested in understanding how we can use the immune system to such an important thing as clearing up the senescent cells and how we can really, and in a way, yeah, I didn't know we can use this as a therapy for certain diseases. Anyway, um, I think we would like as an end of our show, we usually like to ask our guests like a question a little bit outside their research. So Jason, maybe you can go ahead.
Yeah, we always keep it fresh, right? We don't go senescent ourselves. So, and the question for day is, what do you think is the biggest misconception in science you would like to resolve? If you could wave your wand and fix it in all of our brains.
So I interpret that question as what is the biggest misconception of science in the public domain? And my view was I lived in Los Angeles for 12 years where I had my lab at UCLA, and we had often encounters with the Hollywood crowd, and my neighbors were part of the Hollywood industry and names you would recognize, but I won't say it. But and we would have occasionally people come into the lab, major producers and things, because they felt donating some money or whatever it was. We were just show ponies at that point. But the problem I had is the depiction of scientists in Hollywood, and this is to me the biggest misconception is, and I told this to people, but there's always this association of mad scientists, which is completely nonsense, but somehow that's always permeated into Hollywood. And also, if anyone has ever done any time in a lab, being a postdoc or a graduate student or something, the amount of drama and dynamics that goes on in a lab could really be a basis of any sitcom that you see. So this is where I think scientists as normal as human beings should be more portrayed in Hollywood, and that's the biggest misconception that I have a pet peeve.
I would definitely watch that sitcom, the drama in the lab. I definitely think it's about time that they do something of that sort. You should just
I,
use your contacts
I know
for
I
that.
don't know what your podcast outreach is, but Hollywood, if they're listening, I can join into screenwriting.
And as the last thing, any positions at your lab or maybe at your company that you would like to just advertise, feel free. This is the moment.
Oh yes, I'm always looking for um, postdoctoral fellows. And I just got an email yesterday from someone who was supposed to come next month, but decided that they don't want to stay in academia. And this is a problem we all encountering a lot. And but the I can speak as myself. My postdoc years were absolutely, the time when you don't have to worry about money and you can just build hypotheses and spend money like crazy. And it's the PI's job to bring the money in. So this is a good time to spend time in the lab. So postdoctoral fellows, I don't look for any particular training, but just curiosity of science and contemporary molecular biology techniques, and that's my I would love to get if someone responds to this, that would be great.
Well, there you go. Anybody interested, you can check out Anil Bhushan's lab. Also, his latest publication at Med about the INKT cells and senescence. And thank you so much for joining us. It's been a super interesting conversation.
Thank you for
Thank
having
you.
me.
It was fun.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and roundup papers. You can also reach out to us on Twitter at @immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.